Bonjour tout le monde, bon appétit. Donc, merci pour assister à la conférence de Dr. Tanya Robinson. Tanya Robinson a fait son doctorat à l'Université de Cornell et maintenant elle est postdoc. Et j'avoue qu'elle n'était pas très productive pendant son doc. Pendant 6 ans, elle a publié seulement 12 articles. Dans Nature Planet Science, mais 12 seulement. Et donc, mais ici, il va nous raconter l'histoire des Bryophytes. Donc, merci beaucoup. Thank you very much for accepting the invitation.
Yeah, great to be here. So is this on or? Okay, great. Okay. Cool. Oh, let's see. Let me just reset there we go. Okay. Well, I hope you said nice things because I didn't understand any of that. So, um, all right. So, yeah, today I'm going to be talking to you about hornworts, which, Juan Carlos is sort of, one of the big hornwort experts in the world, and this is how I got to know him. And specifically, they have an organelle in their chloroplasts that called a pyrenoid. But before I can really tell you about the pyrenoid, I need to tell you a little bit about Rubisco. So, Rubisco is the carbon-fixing enzyme. So it's, essential for photosynthesis, and I think this is something that most biologists know. But it's worth taking a second to sort of pause and think about because, beyond just being important for supplying carbon for photosynthesis, it also has major implications on global primary productivity, as well as the various food products that we rely or plant products that we rely on, like food and lumber, and even the much older plant products that we rely on as well. All of the carbon in all of these things, including you, all of the carbon in these things was first fixed by Rubisco. So it's a kind of mind-bogglingly important enzyme, that has major global, implications. But despite its importance, Rubisco is sort of known to have a few problems. The first problem is that Rubisco is slow when you compare it to other enzymes. So it's catalyzing between 3 and 10 reactions per second, which doesn't sound too bad until you compare it to something like a carbonic anhydrase, which is catalyzing about 1 million reactions per second. Plants have a pretty straightforward solution to this issue, and that is that they just make lots of Rubisco, so much so that it's considered the most abundant enzyme on Earth, and its global mass is estimated to be somewhere around 0.7 gigatons. And so, this is a nice, quick solution, but it also incurs a major cost to the plants, and that is, it's expensive to synthesize all these proteins in terms of nitrogen. And then the second problem is that Rubisco actually, it has a hard time telling the difference between oxygen and carbon dioxide. So in addition to this carboxylation reaction, which sort of sustains, life on Earth, Rubisco also catalyzes the oxygenation of our UBP. And this results in a 2-carbon sugar called 2PG, and the plant actually can't really use the 2PG, and so it has to recycle it. And this is done by a metabolic pathway that we call photorespiration. And photorespiration results in the net loss of a previously fixed CO2 and requires the expenditure of energy. And so, and to sort of put it into perspective, in a typical C3 plant, this is happening about a quarter of the time. About a quarter of the time, it's mixing up CO2 and O2, and so this can be quite costly for the plants. And it's led many in the field to conclude that photosynthesis is often limited by Rubisco, and this is especially true as temperatures go up, because it has an even harder time discriminating between O2 and CO2 as it gets warmer. But there's a fairly straightforward solution to these limitations, and that is to simply increase the concentration, the local concentration of CO2 around Rubisco, so that the active sites are saturated, and it's much less likely to make this mistake leading to photorespiration. And so broadly, this approach is called a CO2 concentrating mechanism. And many will be familiar with, you know, the biochemical CO2 concentrating mechanisms that we see in things like C4 photosynthesis and CAM photosynthesis, where there's an intermediate enzyme that is fixing the carbon, and then those sugars get shuttled to a Rubisco-rich environment, and then the CO2 actually gets re-released and then fixed again by Rubisco. Um, um, but people are often less familiar with these biophysical CCMs, and these are found in uh, cyanobacteria in the form of carboxysomes and in purinoids of sort of some eukaryotes. And so today I'm going to be talking pretty much for the rest of the time about purinoids. Um, and how, you know, I guess like at a high level, what is a purinoid-based CO2 concentrating mechanism? What is a PCCM? Well, first, you have to assemble a purinoid, and what you do is you take Rubisco that is normally diffuse in the chloroplast, and you condense it into a matrix. And then the second step is that you deliver CO2 into that matrix. And that's it. So conceptually, it's very simple. And I think uh, this has gotten people really excited about it in terms of its engineering potential because it's so simple. And to kind of give you a sense of how impactful these biophysical CCMs are, consider that half of global net primary productivity is occurring in the oceans. But only 3% of global Rubisco mass is found in the oceans. So they are fixing a lot more carbon per unit Rubisco, and that is, can be attributed in large part to these CCMs. And so there's serious interest in introducing these CCMs into crops. And people have done the modeling. I'm not a modeling guy, so I'm just going to trust them. They say that if you were to put this into a crop plant or a C3 plant like rice, you could expect a 80% increase in daily carbon gain, as well as because you don't have to make as much Rubisco, you're going to increase the nitrogen use efficiency too. And so I've shown you multiple images of this organism. Here's a little cartoon of it. It's the model algae Chlamydomonas. Oh yeah, I have a laser. Let's see. Great. Okay, so Chlamydomonas, it has this sort of U-shaped or a horseshoe-shaped chloroplast, and then this pink blob here in the middle that's the purinoid. And so this has really been the sort of model organism for studying purinoid physiology as well as molecular biology for the past few decades. And it's been immensely useful, but there is a bit of a problem with it, and that is that 1 billion years is a long time, which is about how much time separates our crop plants from Chlamydomonas. And in that 1 billion years, some important differences on the surface of Rubisco have evolved, as well as differences in the structure of the chloroplasts between land plants and algae that make the engineering of a purinoid-based CO2 concentrating mechanism into crop plants a little more challenging. But that's where hornworts come in. So hornworts, they are the only land plant to have purinoids. And so if you're not familiar with hornworts, I forgive you. They are bryophytes, so they're sister to the mosses and the liverworts. Here we have a beautiful SEM of a hornwort chloroplast. And then these blobs that I false colored in magenta are their purinoids. And again, if you're not familiar, this is what they look like. They're rather diminutive, but they're quite cool. They're really beautiful organisms, I think. And so when I started my PhD, people were, starting to claim that this hornwort Anthoceros agrestis was sort of the model hornwort. They started to do the work to establish it as a model species. But there wasn't a genome at the time, although it wasn't too long before actually my PhD advisor, Fei Wei Li, had completed the first hornwort genome. So yeah, but when I started my PhD, there weren't transformation methods in place for the hornworts, but it did seem like it was a really promising, new field, and a promising new perspective on pyronoid biology, especially if the goal for people is this application, right, of introducing a CCM into crops. And so, yeah, with these high quality genomes and growth media and acenic cultures, I thought this would be a really cool project. And so the first thing I did with this postdoc in our lab at the time, Declan Lafferty, was we worked on establishing a transformation method for the hornworts. And so we used biolistics, which I don't know how many of you are familiar with it, but it's the gene gun. And it's a really pretty simple concept. You basically take gold particles and you coat them in the DNA that you want to transform into the organism, and then you shoot them at the organism, and they'll randomly insert into the genome. And so that's sort of our technique that we use, and it's quite good at getting transient expression. So we can have transient expression in only 3 days. But if you are looking for more stable lines, partially due to how slow the hornworts grow in a lab setting, you have to wait a bit longer. Okay, so before I kind of dive into what I've done, I think it's important to acknowledge that there are some organizational differences between the hornwort and the algal chloroplast and pyronoid. So here we have a hornwort chloroplast. So just like Chlamydomonas, they have just the one chloroplast per cell, which is an exceptional feature among land plants. One difference that they have from Chlamydomonas is that they have these so-called multiple pyronoids. So each one of these blobs is a pyronoid, whereas in Chlamydomonas they have just the one pyronoid in the center of the chloroplast, and then this white ring that is surrounding the pyronoid it's basically composed of starch. It's a shell surrounding the pyronoid. And people have supposed for a long time that this starch sheath is important for preventing the leakage of CO2 out of the pyronoid, but partially because of what we see in the hornworts, I think that's probably not the case. Oh, right. And so then I guess in general, you're going to see a lot of confocal images over the rest of the talk. And I basically just employ a pretty straightforward kind of 2-tag system where basically I'll have my pyronoid marker tagged in M scarlet, so that will signify to you where the pyronoid is. And then my gene of interest will generally be tagged with M venus. So that's just to help you get oriented. And so what is my pyronoid marker? I'm using this protein Rubisco activase, which is just it just helps with the function of Rubisco without getting too much into the details. And it's, you can see right here, I also have tagged, just to kind of demonstrate that this works well, I've tagged the Rubisco as well, and they co-localize really nicely. And then the other thing here is that you can see that the blue is, this is chlorophyll autofluorescence. So this is just to show you where the chloroplast is in relation to the pyronoid. Okay, so yeah, as I already indicated, one of the first things to notice about the hornwort pyronoids, as opposed to Chlamydomonas, is that they have these multiple pyronoids instead of just a single pyronoid per chloroplast. So this is a Z-stack. We're going down through the hornwort tissue. And so, I hope what you can see here is that there are multiple of these condensates scattered throughout the chloroplast. And so this obviously, if you're going to be delivering CO2 into a pyronoid, this changes how these various components that might deliver the CO2 would be organized. And then the other thing is that the hornworts actually don't have a starch sheath. So again, as I said, people really thought that this starch, or think that this starch sheath is really important for preventing the leakage of CO2 out of the pyrenoid, and it could be, but there's actually not really great evidence to support that. And on the contrary, the hornworts seem to have a CCM that works just fine, and they don't have any starch sheath. So it may not be all that important for the delivery of, or for the preventing of leakage of CO2 out of the pyrenoid. And then, well, the colors look not great. Okay, so now hopefully you're a little bit better oriented in terms of the hornwort chloroplast. So I'm just going to make I have here like a little cartoon of a hornwort chloroplast, and we're going to kind of walk through various components and kind of fill things in to try to make sense of spatially how the chloroplast is organized. So here in magenta, these are the multiple pyrenoids of the hornworts, and then these wavy guys, these are thylakoids, which are surrounding the pyrenoids. And so what have been my big questions over the past few years? Well, you can probably figure them out. How do hornworts deliver CO2 to their pyrenoids? And how do hornworts assemble their pyrenoids? And I'll start with the delivery story first, mostly because the second bit is, I think, more complete and more interesting, and everyone likes to finish a talk strong. So yeah, we'll talk about this delivery of CO2 first. And so hopefully, I swear the colors look better on my screen, but hopefully you can see it okay. But so how does the Chlamydomonas CCM deliver CO2 to its pyrenoids? Well, CO2 is a non-polar gas, so you can imagine this is not actually an easy thing for an organism to move around. But fortunately, when CO2 dissolves in water, it exists in equilibrium with bicarbonate. And so this is something that a cell can work with. And so the Chlamydomonas takes advantage of these intercompartmental pH differences to sort of manipulate the inorganic carbon and move it into the pyrenoid. And so they will import bicarbonate from the cytosol and the environment, the extracellular environment, and they'll pump it into these specialized membranes called thylakoid tubules, which poke through the pyrenoid and allow and then the bicarbonate can be delivered into the center of the pyrenoid, converted to CO2, and that enriches the pyrenoid in CO2. And so it's been found that there are basically 5 proteins that constitute the CO2 delivery system. This is an oversimplification. But so they have these inorganic carbon transporters, these bicarbonate transporters, which move bicarbonate into the chloroplast. And then once in the chloroplast, there's yet again another inorganic carbon transporter, this bestrophin channel, which allows the delivery of this bicarbonate into these thylakoids. So this bestrophin channel is one of the first important proteins that I want to recognize. And then there in the center of these pyrenoids is this carbonic anhydrase 3. This is localized specifically, I hope you can see here, to the center of the pyrenoid. And so, the conversion of bicarbonate to CO2 is a spontaneous reaction, but it's a slow reaction. And so this carbonic anhydrase 3 can catalyze that conversion to CO2 to help the delivery. And then finally, another carbonic anhydrase is called the LCIB. And this, in Chlamydomonas, it depends on the context, but for simplicity's sake, LCIB sort of forms this ring around the pyrenoid, which you can see here. And it's preventing So basically, if CO2 were to leak out of the pyrenoid, there's a carbonic anhydrase that's right there, and the pH in the stroma is such that it would get converted right back to bicarbonate, so that it wouldn't leak out of the chloroplast. The cell had just done all this work to bring it into the chloroplast. So and this sort of is what I think constitutes, sort of a minimal CO2 delivery system. There's probably much more going on than this, but this is what we know so far. And so, yeah, when I started my PhD, I without the ability to make mutants yet in the hornworts and CRISPR, I wondered how, I could kind of figure out how to identify what these CO2 delivery components are in the hornworts. And it actually turned out to be a lot easier or like more straightforward than I expected. So I started by just looking at all of the carbonic anhydrase families in across land plants, including the hornworts. I started looking across anion channels found in the chloroplast as well. And to make a really long story short, basically what I found is that the hornworts don't have any orthologs to these first two inorganic carbon transporters. These, at the plasma membrane or at the chloroplast membrane. But they do have an ortholog to this bestrophin channel, but so do other many other plants. And they have an ortholog to this carbonic anhydrase 3, which is only found in bryophytes and green algae. And they have an ortholog to this LCIB, which is only found in the algae and in the hornworts. And so this sort of conservation of these three proteins in the hornworts, but the incomplete conservation across the rest of the land plants led me to suspect that these might be responsible for what's delivering the CO2 in the hornworts. And so first I looked at the localization of this bestrophin channel. And so you can see it's sort of, it's distributed across the whole chloroplast. But hopefully, if I take away the pyrenoid channel, what's clear is that it's actually more intensely concentrated right in the periphery of the pyrenoids. This was really cool. Oh, oops, animation's in the wrong place. Okay, there it goes. It's on the thylakoid. This is really cool because, oh, okay, well, I apologize. There's supposed to be another image here, but this is the same localization pattern as in Chlamydomonas. Chlamydomonas, the bestrophin channel, also localizes right in the periphery of the pyrenoid. So this was a kind of a surprising parallel. And then got to go through the animation again. Okay, and then when I looked at the carbonic anhydrase 3 localization in the hornworts, what I found this was one of my first early exciting results was that this carbonic anhydrase 3, it localizes to the interior edge of the pyrenoids. So these pyrenoids are organized in a sort of cluster, and then there's this sort of hollow shell in the middle, and the carbonic anhydrase 3 is lining the inside of that shell. And so again, if I take away the other channels, maybe it's a little easier to see. It's a very distinct localization pattern. And so, on our little cartoon, it goes somewhere like that. Oh, and see, there's the correct one for this one. So yeah, again, it's a parallel kind of localization pattern, but not exactly the same. And then finally, there's this LCIB, which again is not present in the rest of the land plants, and this was one of the proteins that I was most excited about when I started working on this stuff. But actually, in the hornworts, it localizes in a way that's quite different from the algae. So it localizes right to the edge of the chloroplast envelope. So this, so you can see this ring here of the LCIB, and then TIC40 is a known envelope protein, and they co-localize basically perfectly. And so there's the whole chloroplast. So this LCIB is localizing right to the edge of the chloroplast membrane. Which maybe at first blush, based on what I've told you about Chlamydomonas, doesn't make as much sense as the other two localizations, but I think it will in just a second, hopefully. Okay, so if we put it all together and try to do a little synthesis here, maybe we can make sense of what's going on here for the hornwort CO2 delivery system. So it seems that just like in algae, they have this bestrophin channel which can conduct bicarbonate into the thylakoid membranes. And then that bicarbonate can be, once it kind of arrives at the central location in the chloroplast where this carbonic anhydrase 3 is, that can catalyze its conversion to CO2, which will enrich the adjacent pyrenoids in CO2. And then similar to the supposed function in Chlamydomonas, this LCIB could function to recapture CO2, which might leak out of the pyrenoids. But I actually think that if we consider the context that hornworts, live as when we kind of in contrast to algae, I think there's an alternative explanation for this localization, which makes more sense, and that is that hornworts live on land as opposed to the algae, which are in an aquatic environment where bicarbonate is going to be the major species of inorganic carbon available to them, and so instead of importing bicarbonate from the environment, what they're doing is CO2 is diffusing into the chloroplast, and if LCIB is localized right to the edge of the chloroplast, then it can convert it into bicarbonate as soon as it enters and sort of trap it in the chloroplast, presumably. So this is something that will have to be tested down the line. And then again, if we consider the context in which hornworts live, they're not aquatic, and so they wouldn't have these various inorganic carbon transporters to bring in extracellular bicarbonate. So it's this is right now a working model. It's a hypothesis. There's a lot of work that needs to be done to sort of validate it, but I think that it's a relatively plausible hypothesis. Yeah, it seems that the hornworts have conserved this CO2 delivery system relative to the algae, and I think that this early look at the hornwort chloroplast and CO2 delivery system gives us some important design cues if we're going to try to engineer crop plants with PCCMs. Maybe you don't need these starch sheaths, and maybe you don't need to worry about getting these special thylakoids poking straight into a pyrenoid matrix. Maybe you can just get the carbonic anhydrase around the pyrenoid, and that's good enough. Oh, yeah, I wasn't sure I'd have a laser, so, but again, further work is needed. Okay, so yeah, that's the first story. It's an interesting start, but it is a little bit on the incomplete side of things. So now let's look at the next story, this pyrenoid assembly question. So yeah, how do hornworts assemble their pyrenoids? Well, once again, let's go back to Chlamydomonas and look and see how chlamys does it. So in chlamys, there's this protein called EPIC1, which is when they first identified it through the many Chlamydomonas mutants. And what you can see from this image here is that this EPIC1 mutant, you still get the starch forming in this little cluster here, but there's virtually no pyrenoid. There's no condensate. And so that was sort of the first clue that maybe this has something to do with pyrenoid formation. And so what is the EPIC1 protein? Well, it's an intrinsically disordered protein, and it follows a sort of sticker spacer model, which, hopefully this little cartoon makes it clear. Basically, it's a protein which has these highly disordered sequences that are super flexible, and then it has these little stickers which will bind to a certain thing, in this case, Rubisco. So by having multiple stickers on a protein, it enables the protein to sort of link its target together into a matrix, and in this case, form a pyrenoid. It's not exactly true, but it's sort of true. This EPIC1 is incompatible with the land plant Rubisco in that it can't directly bind to the native plant rubisco. It really is quite specific to the climate, well, chlorophyte rubisco. But some folks have done some work where they basically expressed the algal small subunit of rubisco, with EPIC1, and they were able to form these condensates. But it's a slightly more complex engineering challenge to do this. But anyway, so when people started to get more and more interested in kind of pyrenoid biology, in the formation of pyrenoids, they started looking at across other organisms beyond Chlamydomonas, and there seemed to be a sort of a rule forming. Everything that had been identified up to 2024 followed this sticker spacer model. And so there's this algae chlorella, and it also has these many rubisco binding stickers, but they bind to a different part of rubisco. And this is also true for this diatom. And so this is kind of a striking pattern across over 1 billion years of evolution, and it suggests that maybe there's only one way to make pyrenoids. And I think this was something people were starting to think. But so when I started looking for the linker in hornworts, that's exactly what I thought too. I started looking for these intrinsically disordered linker proteins. And to make another long story short, I tried a lot of things, and nothing worked. I could never find this intrinsically disordered protein, and sort of had to go back to the drawing board and reconsider things. And so if we take a step back and look at rubisco one more time, rubisco is a so-called hexadecamer, so it's composed of 8 large subunits and 8 small subunits. And the large subunits are where like the actual catalysis occurs, and the small subunits sort of stabilize the enzyme. But there are these proteins are deeply conserved across plants. I was sort of struck when I was looking at an alignment of the small subunit in hornworts, well, across all of the plants. And what I found was that our model hornwort Anthoceros agrestis has this C-terminal extension on the small subunit. It's about 100 amino acids, which is, it stuck out like a sore thumb. Like it's massive compared to, the variations that you see. So like, from Chlamydomonas to Arabidopsis, the length of the small subunit of rubisco is probably within 10 amino acids of each other. And so this, extra length was compelling. And it was also abundantly expressed. I did at first I thought it was fake. I did all my double checking, and it, is a valid gene model, and we even had good sort of proteomic support for that it existed. This got more exciting when I realized that this weird RBCS seems to correlate with the presence of pyrenoids. So this whole time I've been telling you that hornworts have pyrenoids, but not all hornworts have pyrenoids. So there's actually a close relative of our model hornwort Anthoceros effusiformis, which doesn't have this weird rubisco small subunit, and it doesn't have pyrenoids. And so this was sort of a kind of an interesting correlation. So here's an image of both. And so, yeah, sort of the first thing I wondered is, okay, well, what does this weird C-terminal extension do? So the full length RBCS sequence, and for now, I'll just call this the RBCS asterisk or RBCS star. We don't really know enough about function to give it a name. And so it unsurprisingly localizes to these pyrenoids, but it also forms all these extra little puncta in the cell too, which is sort of the first indication that, okay, this has some sort of propensity to self-aggregate. And so then the next thing I did was basically I chopped off this RBCS, and so now I'm just expressing this C-terminal extension in the chloroplast just the weird bit. And it still localizes to the pyrenoids. And it also forms all these additional puncta in the chloroplast too. So again, demonstrating that this has some sort of propensity to self-aggregate, and that it has, affinity for, what is, whatever is already in the pyrenoid. So yeah, this is another great hint that, okay, maybe this is potentially our linker protein in the hornworts. But it's super cool, but this doesn't say that it's causing pyrenoid formation, right? But as I showed you earlier, we have sort of a very straightforward natural experiment that we can conduct, that is basically suggested by the natural diversity of the hornworts, and so that is, if you express this RBCS sequence into Anthoceros fusiformis, the one without pyrenoids, can you then get pyrenoids? And so that's what it did. So to start, this is what the native RBCS in that organism looks like. So again, it doesn't have pyrenoids. It doesn't have this strange RBCS, and so you can see that the Rubisco signal is diffused throughout the chloroplast, unsurprisingly. But when you express it with the here in magenta, this RBCS star, you can see that now the Rubisco is condensed into these. They're not quite as dense as what you would see in the Anthoceros agrestis pyrenoids, but they are, the condensates for sure. And so, this was a really compelling, result. And, it suggests that the hornworts are sort of breaking the mold. They have, instead of this intrinsically disordered protein, they have an innate built-in linker protein, which is making the Rubiscos oligomerize with each other. And so, we wanted to see whether this was really true, and so the best thing to do is to express it into a plant system where there's definitely no pyrenoids, and it's super well established. So we collaborated with our friends Alistair and Yue at the University of Edinburgh, who are great in terms of they're big in the world of CCMs, but they're also just great plant biotechnologists, and so they expressed this. So first of all, the native RBCS in Arabidopsis is diffused in the chloroplast, just as you would expect, and here's a TEM to sort of complement that. And when they express the RBCS star into the chloroplast, you can see that these condensates form, just like in fusiformis. And so this was, a super exciting result. And they took it one step further by just fusing this native C-terminal extension to the native Arabidopsis RBCS, and it still forms those condensates. And so that, maybe doesn't seem that, striking, but what it shows is that there's nothing special about the hornwort RBCS. It is this C-terminal extension that is doing the work here in terms of condensing the Rubisco. I think even the cherry on top of this was that when they expressed this in their mutants, which this mutant 1A, 2B, 3B, it basically has all of this native small subunit knocked out, and so that's why it's pathetic. When they expressed the RBCS star into it, it complemented it more or less. So this is a functional Rubisco small subunit. And so this has significant engineering potential because, as I said before, these other algal linkers have so far proven to be incompatible with the land plant Rubisco. They're binding to algae-specific surfaces on the protein. But the hornwort ones, it seems to be agnostic to the whatever it's being expressed in, because it's only interacting with spoilers itself, and also it means that if you wanted to use this to engineer a pyrenoid into a crop plant, you needn't express a foreign RBCS as well. You could just modify the native RBCS of that organism with this C-terminal extension, and it would do the trick. But it's cool to know a thing works, but I think most of us in this room will want to know how a thing works. And so the next thing I did was sort of drill into the actual function or mechanism that's going on here. And so if we take a closer look at it, now I'm brave enough. I've already sort of given it away, but I'm calling it RBCS star sequestration associated region. So this is our star region. This is that C-terminal extension. And the C-terminal extension can kind of be broken up into 3 subdomains. There's this unstructured bit here, which I think just gives it sort of conformational freedom. And then there's these helical bits here, which are sort of these motifs. So if you align them based off of their kind of amino acid properties rather than just the letter, you can see that there's, especially in motifs 2 and 3, there's a clear pattern of a sort of motif within each of these regions. And so, these motifs were sort of suggestive of a potential, maybe they have a potential function. And so the first thing that I did was make a bunch of truncations of this star to try to get a sense of if these truncations would affect the localization. And so by removing the most C-terminal motif, you can see that the protein still localizes to the pyrenoid. And likewise, if you were to remove the second motif but keep the most C-terminal motif, this motif 3, it still localizes to the pyrenoid. In both cases, though, you still get these little puncta that form, like in the full-length sequence. But if you chop off both motifs 2 and 3, the signal becomes much more diffuse throughout the chloroplast. It's still showing some preferential localization within the pyrenoids, but not nearly as much. And this is sort of just a quantification to make it easier to see that. But again, we phoned a friend because looking at these things within the hornwort system, is complicated because they're already expressing the star. So getting at function or mechanism is a little tricky. And so they did the same thing. And so this is just a condensed information, but basically they found the same thing. You can express the full-length RBCS star, or you can express the truncations of motif 2 or motif 3, and you still get nice, clean condensates forming. However, if you remove both motifs 2 and motif 3, it abolishes the formation of these condensates. So these are, appear to be very important for the function of the condensation. And they once again, they took it one step further. They also did a truncation where they removed this unstructured bit, and they found that these condensates still form, but you can see that they're quite weird now. Rather than being almost spherical, they're kind of smushed all over the chloroplast, and they don't seem to get to quite the same size. And so this, and here's a quantification of that information. And so this suggests that these motifs motifs 2 and 3 are likely redundant. Whereas motif 1, it may have some affinity for the pyrenoid, but as we saw, it still can localize to the pyrenoid in the hornworts. But whatever affinity it has is too low to work on its own in a system without pyrenoids already in place. And so when I looked closer at the sequences for these motifs, I realized that these motifs 2 and 3 are like canonical coiled coil sequences. And for the uninitiated, a coiled coil is a pretty straightforward protein motif where it's basically just an ambipathic helix. So one face of the alpha helix is hydrophobic, and there's a certain kind of specific geometry that I don't really want to get into, but it basically forces these helices to wrap around each other, hence the name coiled coil. And they have really well-established rules. It's one of the most well-understood protein motifs out there. We'll click. Oh, there we go. Okay, and so because it has these really well-established rules, it's actually really easy to break them. And so here we have, sort of this. This is motif 2. This is motif 3. And so, you have these hydrophobic residues that are important for the oligomerization of these coiled coils. So if you were to just change those hydrophobic residues to something that's more hydrophilic, you might abolish the actual oligomerization of the protein. And so that's exactly what we did. So this mutant here called 6q, and basically each of these hydrophobic, half of the hydrophobic residues on each motif were mutated to something hydrophilic, in this case, glutamine. And so this is, so the protein was then purified and ran on size exclusion column. And so, and I did modeling the modeling basically said that the RBCS star coils will form a tetramer. And so it was really cool to see when we or when we ran this protein on a size exclusion column, it eluted at a volume that was consistent with a tetramer. And so with size exclusion size exclusion columns, basically larger complexes, larger proteins, they elute first, and the smaller ones elute later. And so when we did this, these mutations, what we found was that the 6q mutant eluted at a volume that was actually consistent with a monomer. So, these few mutations were sufficient to abolish the coiled coil interactions. And we also did a cross-linking where these lysines here were cross-linked. And you can see here on the gel, I'm sorry, I know I'm out of time. So you can see here on the gel that the uncross-linked, uncross-linked are sort of, they're running at a size that is consistent with a monomer on an SDS page, which makes sense because SDS pages are denaturing the protein, whereas the cross-linked ones are running at a size that is consistent with a tetramer. But this isn't the case for the 6q mutant. Again, establishing that this is based off of a coiled coil interaction.
Dígame.
Okay, and so I am, when I got to this phase of the project, I was right at the end of my PhD, and I'm, at the time I really didn't know anything about protein biology, protein chemistry, and so I just had an idea, and these people really made it possible. And so, yeah, I have to acknowledge all the help that I had here. But so this is sort of our hypothesis then for how the hornworts are forming their pyrenoids. So these stars, which are integrated into the Rubisco holoenzyme, are oligomerizing with each other in a tetramer through these coiled coil interactions. And actually, sort of on for me, the most exciting photo that I've
Bolivia y norte
seen is
y en Venezuela.
yeah. Can you sorry, can you mute yourself?
Pero esas no las tenemos nosotros porque nosotros bajamos esos datos.
So on a cryogrid, these Rubiscos, you can see that they're linked together by these thin filaments, which we hypothesize
No,
are
esa no nos sirve.
stars. So,
Pues no sé, es que es como, bueno, pero también
Alejandra, you're not muted. Okay, hopefully that worked. Okay, so in summary, these this RBCS star thing is pretty neat, super cool. So it's a so-called innate linker. It's built into the Rubisco rather than being another secondary protein that is mediating the formation of the pyrenoids. It's a functional RBCS. So this additional domain doesn't affect the ability of it of Rubisco to fix carbon. And it's a so-called universal linker, which is a big has been a big goal for the CCM community for a long time, or for a number of years, and so people are quite excited about that aspect of it. And it functions by a really simple, well-understood motif, these coiled coils. And so this, I think, presents really interesting sort of synthetic biology approaches. You can manipulate these coiled coils to potentially alter the behavior of this protein and the sort of the properties of the condensates, properties of the pyrenoid. So it seems that, at least I have a nice hypothesis for how this CO2 delivery works. Maybe it seems like this whole pyrenoid assembly story is all wrapped up. So that's cool. I guess I can retire. But maybe not. So the next thing that I want to work on, which I'm really excited about, is, again, I've been telling you hornworts have pyrenoids, and we've been talking about this RBCS star thing. But it actually turns out that the pyrenoids in hornworts, based off of work from Juan Carlos in 2012-ish, the pyrenoids have evolved multiple times across the hornworts, and this actually is kind of reflected in the different ultrastructures that you can see among the different hornwort lineages. And so, in the case up here, I've used this Anthoceros fusiformis and Anthoceros agrestis as a nice, tool to sort of pick apart what this linker protein might be. And this is important because actually, most of the other hornworts don't have anything like RBCS star, and so they're presumably condensing their pyrenoids in a totally different way. So there's, I think this is a really exciting system where you can look at, this relationship between, from all the way from sequence up to sort of physiology. You can look at how different proteins either kind of neo-functionalize or de novo evolve in order to produce a trait that is important to the plant. And so, yeah, I want to look across these many independent origins of pyrenoids in the hornworts to try to understand how these linker proteins evolve. But I think it's also interesting, too, you have all these lineages without pyrenoids. And so by looking at the localization of this so-called CO2 delivery machinery, can we understand how the CO2 delivery machinery evolves, too? And so, yeah, it could be that there are all different kinds of linkers found in hornworts, and maybe there's another third thing that we haven't imagined yet, which I think is kind of cool. And this is all something that I think is quite doable because actually our lab has genomes for most of these organisms and cultures for all of them here. And the transformation, it's a gene gun, so it'll work on all of them, but we've only verified it with these ones. So I think this is a really tractable and exciting system to understand something so cool as a pyrenoid assembly. And so with that, I just want to thank everyone who made this project possible, as well as the various funding that we've had over the years. And yeah, again, I want to thank our collaborators who really helped make this possible. And I'll take your questions.